This story started maybe 10 or 12 years ago when my PhD advisor sent me to the desert and said, "Oh, just go check it out and see what you can find." And while working under the guise of a behavior experiment, I secretly became obsessed with this question: how do desert animals survive? And so how I came about this question was basically by just poking around. So I walked through this kind of amazing landscape, and I look under rocks and under like felled logs, and even though the environment was terribly inhospitable, at least to me, everywhere there was just life, right? So it was really cool. So you might be kind of sitting in the audience right now wondering like, well, what does this matter to me? I'm in Portland or wherever you're from, and it's not the desert, right? And I would argue, of course, that it should matter for everyone, and basically for the reasons I'm presenting here. So this is a figure from the IPCC 2007 report on climate change, and the picture that it paints is pretty ominous. So what I'm saying here is that what they are saying is over the next 100 years, our world is going to be getting hotter, right? In fact, in some places in North America, we're talking about as hot as, or an increase as much as 3 or 4 degrees centigrade. So that's quite a lot hotter. In addition to being hotter, it's also going to become quite a lot drier. And here the effects are a little bit more regional, centered on the Southwest and some other areas, but still maybe as much as 10 or 15% drier. And so I would argue, just like Rachel had argued a couple of talks ago, is that if we are to understand how animals are going to be adapting to this increasingly harsh, dry, hot climate, maybe we should look at animals that are already adapted to these types of environments. So like I say, for the past maybe 10 or 12 years now, I've worked on this this organism here, Peromyscus eremites, the cactus mouse. It is absolutely the cutest study organism ever. But aside from being really cute, it also offers some what I think are pretty cool opportunities. So this is an animal that in the field lives maybe 6 or 8 months or maybe a year, during which it may never drink water in its entire life. So that's automatically pretty cool. We as mammals are typically pretty water dependent, not so much this animal. Of course, there's no water, so that kind of makes sense. In addition to perhaps never drinking water, it may never urinate. So there's some other kind of another set of interesting questions to be had there. So let's see here. So a couple of years ago, maybe 3 or 4 years ago, when I took my faculty position, one of the first things that I did was I constructed this environmental chamber here. So this is a walk-in environmental chamber, just like a freezer or a fridge that you may have in your labs, but instead of kind of cold, it's kind of hot. So the chamber, we can regulate basically all of the environmental conditions that a mouse, well, maybe not all, some of the environmental conditions that a mouse might experience. In particular, the environmental conditions that we manipulate are things like temperature and relative humidity, as well as rainfall. We can give them water or not. So this allows me I think the really unique opportunity to have animals in somewhat of a native environment, but still kind of do the laboratory-based experiments that one would want to do when investigating things like desert adaptation. So when I first kind of got the mice and they were breeding and everything was great, I did this very simple yet, I think, informative experiment. And so what I did was I took a group of animals. These were sexually mature adult animals, and I deprived them of water for a period of 3 days. So to back up maybe one step, these mice will drink some water if you give it to them, and so the baseline condition is to give them just a little bit of water. When you deprive them of that water over 3 days, well, they lose on average 23% of their body weight, which is quite a lot of body weight if you think about it. You losing as a human being a quarter of your body weight, that's quite a lot. And so this kind of distribution is plotted here in this violin plot, where the y-axis is the percent body weight, and so you can see the average is about 23%. There are some animals that lose a tremendous amount of body weight, as much as 40% or so, and then some animals that lose as little as 10 or so percent. This is actually an amazingly scary experiment to do, because when I went into the kind of mouse room after the 3 days, I said, oh my God, these mice have lost a quarter of their body weight. I thought they were almost dead or whatever. It was bad. But in fact, these mice, even though they've lost as much as in some cases 40% of their body weight, they're basically doing the kind of normal mouse thing. So they're still hopping around, mating, playing, kind of doing all those normal things. So the mice seem to endure this, this really kind of extreme dehydration pretty well. So just to provide some context to what would happen with humans. So humans, of course, we're, like most other mammals, we're exquisitely sensitive to changes in our hydration status. And when we exhibit something like less than 3% dehydration, well, we're thirsty. This is like when we walked to lunch yesterday in Portland. But we have generally mild or no ill effects. When we're between 4 and 10% dehydrated, well, we have a decrease in our cognitive performance, cognitive and physical performance. We may have an acute kidney injury. Our kidneys aren't functioning as per normal, and we have electrolyte derangement. Between 4 and 10% dehydration, you're usually like in the hospital, getting IV fluids, stuff like that. You're not particularly happy. Between 10 and 15%, you have arrhythmia, cardiovascular collapse. These are people that are in the intensive care unit. They're very sick individuals. And of course, greater than 15%, well, as humans, we basically die at about that level. The mice, we're talking about as much as 8 or 9% on average, more than that, and they're kind of just fine. So this is a pretty cool phenomenon. To begin to study this, we decided to look at the kidneys. This seems like an obvious place to start to look because the kidneys have been broadly implicated in fluid and electrolyte balance, right? Like they're the organ that kind of manages fluid, one of the organs that kind of manage the fluids. And previous studies have looked at this type of thing in the Peromyscus system, and they've looked at the structure of the kind of functional unit, the nephron, and what they found is that this loop of Henle, this kind of elongated loop structure here, is very elongated, things like Dipodomys, the other kind of classical desert organisms. And so when we started the study, we did the kind of histology work that would be required to know this. We thought kind of for sure we'd see this elongated loop of Henle, but we didn't. What we saw was basically a kidney that looked like, mouse or kind of any other kidney. There wasn't anything particularly special about the cactus mouse kidney. We kind of weren't convinced. We thought that, there must be something here, right? So we decided to do an RNA-seq study. And so I apologize for the kind of slightly wordy study, but I just want to lay out the details of the studies that forms the basis for what I'll talk about kind of the rest of the talk. So for the experiment, the animals were held in 9 degrees Fahrenheit, 10% relative humidity, and this cycled out a diurnal cycle. So as to say, it got a little bit cooler in the night, a little bit more humid, and then daytime heat was that. These were all adult individuals, both sexes were equally represented, and for 18 of the individuals, they had water just like normal. For the other 18 individuals, they had no water for 72 hours. We generated a bunch of sequence data. We assembled a transcriptome using this protocol that we've developed in the lab over the last couple of years. We use salmon for the quantitation, TX-import to do the gene level analysis, and EdgeR to do the differential expression testing. On top of all of this, for every individual, we have quite an extensive physiology data set, so things like serum electrolytes, blood pressure, and heart rate, a bunch of other things that I kind of won't talk about at all today. If you're really interested, the physiology data is mostly described in this paper from a couple of months ago in Physiology Reports, and the RNA-seq study is in press, but you can see early access in renal physiology. So instead of kind of going through the laundry list of genes and whatnot that sometimes people do with RNA-seq experiments, I want to spend some time talking about some focal kind of hypotheses or whatnot that we have explored. So the first one is related to apoptosis or programmed cell death. And so the idea was here is that if acute dehydration is associated with renal injury, if acutely dehydrating animals is actually bad for them, well, we might see signs of that in the actual kidney. So, surprise, we didn't see that. We saw actually no evidence of apoptosis during acute dehydration. And so let me just take a second here to explain these plots, because you'll see a few more slides of them. So these are split violin plots. And so on the brown side here, this is the distribution of expression values for the dry animals. And on the other side, on the kind of dark blue side, this is the distribution of expression values for the wet or the kind of hydrated individuals. And the y-axis is the log TPM. So more expression higher on the y-axis like you'd expect. And so I'm picking kind of, I'm choosing to show you here kind of 4 of the classic genes involved in apoptotic processes. Working in mammalian genetics means we have a lot of information about how programmed cell death works. So P53, BCL2, BID, CHOP, these are all measures that if apoptosis, if programmed cell death was really, really happening in the kidneys, we'd see differential expression here. But we don't see any of that. So if you can kind of imagine the kind of means, the distributions, they all look relatively same. Well, there's some stuff here, but no significant difference in these genes. So we took a step back and we said, well, maybe the apoptosis thing kind of isn't what we should be looking at. Instead, we should be looking at more synthetic markers of kidney damage. And again, because this is a mouse, we have lots of well-established biomarkers for kidney injury. And here was basically the same thing. So despite the fact that we looked at, I'm showing you here 6, but there are maybe 10 or 12 we looked at in some, none of them showed any sort of differential expression related to acute kidney injury. So no significant differential expression in any of the biomarkers. So that's pretty cool. Humans, we know that acute dehydration is linked to kidney damage, not so in the mice here. So how does kidney injury actually happen? First of all, I told you that 23% of weight loss, that sounds bad. There's no sign of kidney damage. And but what I'll tell you is kidney damage typically results from reduced renal blood pressure. Now, one of the things that you can do is to increase your renal blood pressure, all right? So what happens, imagine you have a hose and a fixed volume of water running through it. If you wanted to make your water pressure higher, well, you would shrink the kind of diameter of the hose, right? Well, it turns out that humans and mice and all that possess really exquisite mechanisms for regulating vascular tone. So without going too much into the biochemistry, there's a really cool molecule. It's a membrane-bound lipid called arachidonic acid. And one of the ways in which it's metabolized is through the cyclooxygenases, and it causes vasodilation. There's another pathway mediated by the cytochrome P450, particularly CYP4A, that produces a molecule called 20-heat, and it causes really, really kind of crazy vasoconstriction. Well, here we see a lot of differential expression. So we looked at all the CYP4As. There are 6 of them expressed in the kidney. 4 of the 6 are differentially expressed in the right direction, meaning vascular tone is increased in the dry animal. 2 of them are not differentially expressed, but their trends kind of head in the right direction. So we interpret this as pretty strong evidence for vasoconstriction, in particular in the kidney in dehydration. So to conclude, we see 23% weight loss during dehydration, no signs of kidney injury, and we really kind of are working on this hypothesis that it's CYP4 expression that causes, via this 20-HET molecule, it causes an increased kind of vascular tone, vasoconstriction, and that kind of regulation of blood pressure that seems to be pretty unique in these individuals is what prevents the actual kidney injury in the cactus mouse. So you might ask, what happens next? Well, we're quantitating 20-HET levels. Because this is a human hypertension pathway, we have both agonists and antagonists. We can manipulate pharmacologically kind of all this stuff. And obviously, we'd love to measure renal blood flow. That's kind of the thing we don't have a great idea how to do that. So looking for a grad student for this project, also a grad student for a funded Dimensions Biodiversity project. So if you're interested in sponges or mice, come talk to me at some point. Thanks, guys.